Let's start with the introduction. In general, influenza known as flu that caused by contagious viral infection that attack the respiratory system. There are 4 types of influenza virus, which are A, B, C, and D. Among all of the viruses, only 3 types of the virus are able to infect human, which are type A, B, and C. Type A and B is a seasonal influenza. Seasonal influenza outbreak caused by a small changes in viruses that are already circulating around human and to which people that have immunity. Influenza viruses exist as a large group closely related viral genome that also called quasi-species. The composition of influenza A viral quasi-species can be determined by accurate and sensitive sequencing technique and also with the data analysis pipeline. Based on the paper, whole genome influenza A quasi-species analysis were compared by using 2 benchtop next-generation sequencing. First technique is the Illumina MiSeq sequencing by synthesis, and another technique is the ion torrent PGM semiconductor sequencing technique. Discussion and result. The aim of the experiment is to compare the performance of 2 throughput sequence instruments and determine the complexity of an influenza A virus quasi-species. The experiment was done by comparing the accuracy and sensitivity of the sequencer using plasmid DNA. The plasmid DNA were generated with 2 tracer mutations in order to determine the sensitivity of sequencer for picking out the occurrence of introduced SNPs. The plasmid chosen for analysis are the ones that allowed to generate PR8 virus with or without the introduced tracer mutation. Based on the result obtained by the researcher, Illumina MiSeq sequencing provide higher quality of trimming and read length is greater than the ion torrent. In silico processing of the sequencing read, Illumina MiSeq lost more base during to quality trimming rather than the short read that often map inaccurately. A few other tests that are done to test the quality and accuracy also shows that Illumina MiSeqs are more sensitive and accurate rather than the ion torrent. Last but not least is the conclusion. The study underlines the power and limitations of 2 commonly used next-generation sequencers for the analysis of influenza gene diversity. They also propose an in silico pipeline for selecting high-quality reads obtained by the NGS platforms. This pipeline is also more widely applicable. Due to the lower total error rate and the higher sequencing quality of the reads, it concludes that the Illumina MiSeq platform is more suitable than the ion torrent PGM for detecting variant sequences, whereas the ion torrent platform has a shorter turnaround time. In addition, we found that the detection limit for reliable recognition of variants in a viral genome require a frequency of 0.5% or higher.